Thank you to the organizer for setting this meeting, which is really very interesting. Especially yesterday, I was quite amazed by the progress that has been made in BMI interface and so on. I'm going to try to give you some overview of the work that we've been doing in our group. We are interested in spatial navigation, and I will try to illustrate some of the differences that may happen in the way activity of place cells is shaped during exploration and during navigation, during goal-directed navigation. So first, a few, let me see if this works, yes. A few basic definitions. So what we call spatial navigation is the ability of the animal to display optimal path in space. And this ability seems to rely on a representation of the environment. The important point that should be noticed is that this representation allows flexible behavior. That is, if the rat or the agent has to change its path based on the knowledge of the relationship between the places in the environment, it's able to take detours or to make a shortcut. So this is a complex behavior, and it's not very surprising that it involves a bunch of brain structures, and this is an illustration of some of the brain structures that we have been studying in our group, with the hippocampus and the place cells at the core of this network. And so today I will speak mostly about place cells, although I will say a few words about another structure, which is the prefrontal cortex, to illustrate the possible coordination or dialogue that may exist between the two structures during navigation, in particular during goal localization. So this is the outline of my talk. I will very, very briefly speak about spatial information processing during spatial navigation. Then I will describe a few findings about place cells during exploration. Before I will try to illustrate some of our findings about place cells during spatial navigation, and then give some conclusion. So there are two ways in which an animal may locate itself in space. When I say animals, we also humans do it the same way. And these two ways are illustrated here. If we look at these two figures, this one illustrates the way that most of us will tend to privilege during our movements in space. We will take some landmarks in space and try to know where we are based on these external landmarks. They may be visual, olfactory, tactile, and so on, but they are mostly external to the animal. The other way, and this is a strategy which we may be using in specific circumstances, is to keep track of our position based on our self-motion, and this relies on the integration of a variety of spatial cues, of self-motion cues, such as vestibular information, proprioceptive information, and so on. So these two strategies are actually complementary, and we'll see that in the natural condition, the agent will be using them alternately and concurrently. The reason why we are interested in place cell in the hippocampus is because of the existence of place cells. So everyone knows that place cells have been discovered almost 50 years ago by John O'Keefe, and the idea of these of John O'Keefe when he discovered the place cells was that they might provide the basis for a spatial map of the environment. These cells are, this is a rat which is recorded in this cylinder, is moving, and if you listen to, I can maybe increase it. Well, if you listen carefully, you will see that one cell happens to fire when the rat is around that location. Okay, now you hear it. And so the idea that this cell, this particular cell, is firing in a given location, and because there are many place cells in the hippocampus, and John O'Keefe was saying that this cell may have different fields, he put forward the theory that these cells might provide the basis for a spatial cognitive map of the environment. So in the rest of the talk, I will show you this type of maps, which is a spatial firing rate map, which show the location where the cell happens to be active in this environment. So what we do, we accumulate activity over a number of minutes during the recording period, and we provide a colored picture which clearly shows that this cell has a firing field in this location in this session. Several properties are important. First of all, as I've said, the main observation is that each place cell has its own spatial firing field, such that if you record, for example, here 4 different place cells, you will have different place field, which, of course, may differ in shape, in size, in location, of course. And this is the overall combination of these cells that provide at any time an information to the rat about its location in space. The second important property is that the spatial firing fields are stable in time. So if you record the same place cell across 2 different episodes, for example, here on day 1 and here on day 2, you see that the field remain quite the same. And the third important property is that these cells are reactive to the cues that I was talking about, the external cues. This is a very classical experiment that has been made in several laboratories, but this one has been made in our laboratory. This is an experiment in which we record the cell with the rat exploring a cylinder arena in a curtain environment, which means that the rat has no access to the external cues from the laboratory, except for one cue that we can manipulate, which is this card, which is here. And what we do is that after recording this cell for one session with the card in a given position, we rotate the card and we record again the same cell to see whether the place field has been affected by this manipulation. And you clearly see that here the field has rotated the same amount and the same direction, of course, than the cue card that was manipulated. Which means that this system is sensitive to external cues. You can manipulate it explicitly, but the point is that if you record also the same cell after you remove this cue card, you see that the field will stay stable for a period of time, which means that this system can also use the self-motion cues, for example, in the dark. And actually, this is something that we do every day when we move in the absence, for example, in the night, in the absence of external cues, we use these external cues, these self-motion cues, I'm sorry. One important property that we have discovered with my colleague André Fenton in the recent year is that even though the fields are stable across session, if you look at in more detail at how the cell will be firing the field during several passes in a single recording session, you will observe that the cell firing may be highly variable across different passes. For example, this is a field of this cell, which has been recorded in a square arena. And you see here two paths through the field which look very similar. And even though they look very similar, you see that the number of action potential that was emitted by the cell is highly different. Here 37, here 5. So if you expect that based on the overall average firing rate of the cell during the recording session, and you compare this observed firing to the expected, you see that there are a huge difference. And we have called this phenomenon overdispersion. It's higher variability, which of course can be measured using normalized index. For example, here the index is almost 1.5, which means that the firing is close to the expected value, while here it's minus 4.2, which means it's very, very far and much lower than the value that was expected. So when you plot these values during a given session, and this is what is done here, you see that there are some values that fall within the expected values, but some values which are higher or lower, which mean that the cell is firing much higher than was expected or much lower than what was expected. Then when you plot this value in such an histogram, you see that this distribution of z-value is departs considerably from the expected statistical distribution for normal distribution. And there is, you can measure this departure by just looking at the variance of this distribution, which is sigma square, here almost 5, which means that there is higher variability than what could be expected based on a traditional statistical distribution. So this overdispersion measure is something that I will return to later in my talk, because it might reflect the strategy that the rat is being using during its movement in space. So now let's turn to navigation. The most classical way of looking at navigation is the Maurice Waterman's navigation task. Here you have a rat navigating for the first, for one of the first trials in the swimming pool. So the platform is here. Here, the same rat after he has learned the task, and you see that he is very, very skillful at finding the platform. And the reason why I'm showing this is first because it clearly shows that the rat is able to display optimal navigation path in that task, but also to say that if you look at a rat with a hippocampal lesion, you will have a rat that behave like this one that was just learning the task, which means that the rat, the trajectory in space will very much look like that. It's not necessary to be a great statistician to understand that this is very different from this. And this is the basis of the O'Keefe reasoning that because there are place cells in the hippocampus and that these lesions produce a very strong impairment in navigation, this structure might be very important for the spatial representation of space. So what did we do with this in mind? First, we asked whether the place cell representation is altered, or in which way it is altered, when you record from a rat solving very simple task, which is a task in which the rat has simply to commute between 2 points, this food dispenser which is here and this food dispenser which is here. This is the W-shaped maze. The rat is simply trained to do that, to get a reward in each of these locations. And what happens when you change slightly the structure of the maze, such that the rat has to adapt to this new configuration, and with two questions in mind. First, does the rat change its path? And that's what we expect if the rat is able to take a shortcut. And second, how the spatial representation is affected under these circumstances. So here is the initial task. This is what we call the shortcut session, in which a portion of this wall has been removed, probably around here. And the question is how this change affects place cell firing. First of all, the rat immediately takes the shortcut, which shows that he has noticed the change in space and adapt to the new topology of the maze. And the main result is about the place cells. Here is a representation of two place cells, one with a field which is far from the location where the portion of the wall has been removed. So the field is here and the shortcut is here. And one with a field which is near the location of the shortcut, that is here. And you see that there is clearly a difference during this session with the shortcut open, with a field which are far from the shortcut, which are mostly unaltered by this change in the topology, while in that case, the place cells happen to stop firing. And this is something which is important because it shows that cells can change locally their firing to update in a way the overall representation of the space which is run by the animal. And this is something that I will return later. Interestingly, there are also some difference between the place cell recorded in CA3 and CA1 and CA3, but I won't develop this aspect later. So now we want to record from place cell during navigation. Of course, it's very hard to do these recordings in a water maze because electricity and water are not good friends. So we have designed a task which is a dry water maze, if I can say it, in which the rat has to explore a cylinder with a card at the periphery of the cylinder. The whole apparatus is surrounded by curtains, so this is the only cue available to the rat to locate a goal, which is here in this figure and here during the solving of the task. So you see that the rat is currently returning to this location. This rat is particular because he is rearing at the goal location, so he clearly shows us that he knows where to go. And once the rat has been waiting for 2 seconds in a row at this location, a food pellet is released from a dispenser at the ceiling. The reward is dropped on the floor, bounced on the floor, and such that the rat has next to explore the cylinder to find the food, to heat it, and to return to the goal location. So this task is, of course, a place navigation task. It's a very schematic and simple task, but yet it has all the requirements for our purpose. It's interesting because first, it allows us to disentangle what's happening during paths oriented to the goal location from those that are used by the rat just to find the food. And it's also interesting in the way that the goal location is not the location at which the rat will find the food reward, such that there is no confounding factor between the goal and the reward. So using this task, we have found several interesting results. And the first is related to what I was talking about, the overdispersion phenomenon. So this is just to remind you that overdispersion is the observation of higher variability than what would be expected based on statistical normal distribution. And what we did is what we compared the overdispersion during simple foraging, during exploration in that task, and during navigation. And what we found is that this measure, which is summarized by these variants of this distribution, is much reduced during navigation compared to simple exploration. So it means that it reflects an active process that is used by the rat during navigation and that is not apparently operational during simple exploration. So to go further, we put forward the hypothesis that it might reflect the use of different kinds of information. And this is to remind you the two kinds of information that we consider. First, the external landmarks, second, the self-motion cues. And so we designed an experiment in which we compared what's happening in the traditional version of the continuous place navigation task that I was talking about, in which both allostatic cues and so external landmarks and self-motion cues, hydrostatic cues are available. There is nothing inherent in the task that forces the rat to use either of these cues. It can use both, to a version in which it is forced to use one kind of these cues because a manipulation of the platform made the hydrostatic cues irrelevant to the solution. So the way we do it is by just training the rat while the platform is rotating and keeping the goal location constant so that it can be located based only on allostatic cues. So what we found under these circumstances is that this is the overdispersion during the version of the task in which both external and self-motion cues are useful. When only allostatic cues can be used, you see that this variance is still much reduced compared to the first version. It means that you can more the activity of these cells can be predicted in a better way based on the overall average firing. So even though it's an hypothesis, we think that this overdispersion reflects the constant shifting of the rat between the use of two different kinds of information, paying attention to the allostatic cues, to external landmarks, and self-localization based on self-motion cues, and occasional resetting of these self-motion cues based on these allostatic cues. Using the same task, we found that the information is important for the spatial information provided by place cells is important for the performance. You can do that just by comparing the performance of the rat, that is the number of reward it gets when it goes to the goal, according to whether the place firing is correctly aligned with the environment or incorrectly aligned with the environment. You see that there is a considerable drop of the performance of the rat. And the interesting point is that if you do the same measurement in a version of the task in which there is no spatial constraint because the rat can locate the goal location based on a cue that is directly put on the floor of the apparatus, so the rat has not to compute its location nor the goal location, you see that there is no effect of the correct or incorrect alignment of the place set. So it means that this system is used for spatial navigation when the use of spatial cues is required. Another important set of observations that we made in this task is that if you look carefully at these maps, which are the maps of 12 different place cells recorded while the rat was solving the task, in addition to the primary field that you see in each of this map, you see that there is a tiny marking of the goal location, systematic, which tends to indicate that the cell, in addition to firing in its primary field, also fire at the goal location. And actually, if you do the classical analysis of looking at that for each cell in a raster in which this each line represents a trial, that is an event when the rat is going to the goal location, that is this location here, you see that there is this tiny and very noisy marks of firing which repeats across trial, and this is the histogram corresponding to this raster. But if you do that at the level of the full population of cells that is recorded, you see that there is a very strong signal that tends to happen here while the rat is at the goal location. Okay, 5 minutes. So the question is, what is this signal for. In a recent experiment with Eleanor Duvel, who was a PhD student in our lab, we made a variation of the task in which we trained the rat to go to 2 different goal locations, and we managed to manipulate the amount of reward that was available at each of these goal locations to address one single hypothesis, that is that the goal-related firing that I was talking about could be related to reward expectation. So the rat was trained to go to these 2 goal locations, and you see here that is okay. Okay, so I'm happy, I'm going there. I received a food reward, which is around here. And the idea was to look at the change in firing at the goal location when you change the amount of reward. So we could either completely suppress the reward when the rat was going to the goal location, or in the contrary, increase the number of rewards available at this goal location. So we observed that the rat immediately adapted its behavior to the change in the reward contingency. This is the performance when there is no change. This is the performance when the rat is in the 1 versus 3 reward contingency. So this is good. And the main idea was to look at whether the goal location was coded in different way during these various combination of reward contingencies. And you see that there is no variation. So even though we don't know what this goal-related firing is used for, we at least can discard the hypothesis that it might be related to the availability of reward, or at least to the expectation of the amount of reward at this location. So it's interesting also to compare this goal-related firing in place cells to the observation that we have made a number of years ago with Vincent Hoch during his thesis, that if you record cells from the frontal cortex, from a prelimbic frontal cortex, you get a number of cells which happen to fire in the goal location, and this firing departs from the firing of place cells because first it's clustered around the goal location, and second, it's much more noisy, and as you can see, the fields are much larger than what is observed for place cells. How long I have? 2 minutes. Okay. So I will skip a number of these slides to get to here. So the question is whether there is a contribution of the prefrontal cortex to the goal firing of place cells, and this is an experiment which we have done with Vincent Hoch, in which we have recorded place cells while the rat was solving this task, and at the same time we would, on some sessions, inactivate the hippocampus, the prefrontal cortex, by injecting lidocaine during the performance of the task to look at whether it would change the behavior and change the goal-related firing of place cells. The results are shown here. First, this activation hardly changed the overall performance, although it produced a small impairment in reaction time for leaving the goal area after the 2 seconds delay when the rat has to stay in the goal location. It doesn't change the goal-related firing. It doesn't change the overall aspect of the local field potential. In contrast, it very strongly changed the overdispersion measure that is, as you can see here, after saline is 3.6, like I was showing earlier, and which is much reduced here. So this result tends to show that there is a modulation of this activity of place cells by the prefrontal cortex, and we suspect that this prefrontal modulation could be a modulation on the way that the rat is shifting between paying attention to self-motion cues and paying attention to external cues. So very briefly, conclusion. So the hippocampal plate cells provide the animal with a spatial map of the environment, which is used for spatial navigation. This relies on both external and self-motion cues. There seems to be a functional interaction between the prefrontal cortex and the hippocampus, and a possible drive of the prefrontal cortex on the place cells. And finally, a set of questions, because I understood that there was some discussion after the talks that are important, at least for me or for us. First, I think it will be necessary in the future to characterize in a better way the difference in firing between exploration and goal-oriented navigation. I don't think that we have all the elements for understanding what's happening exactly during spatial navigation. Second, I think that even though these lab studies are very interesting and provide a number of results that tells us something very important about how place cells might contribute to spatial navigation, they do not reflect the reality. And for me, it will be very important to record place cells in complex natural environments, in bigger environments, such as those that we have seen yesterday in the slide of Kowloon. For example, this tunnel, which are very complex topologically. And another question that we want to ask is the role of the voluntary hippocampus, because all what I've talked about was about dorsal hippocampus. Place cells were recorded in this location, and we still don't know how these two parts of the hippocampus might talk to each other. Thank you very much. This is a slide with people who have participated in various parts of this work. Francesca Sargolini, who is here, Alice Alverne, Eleanor Duvel, Vincent Hock, André Fenton in New York, Pepe Lengsentini, I didn't see him, he should be around, but didn't show up. Bob Muller, who unfortunately died 3 years ago, and Etienne Save in our lab. Thank you very much.